Well, as Hiro said, I'm working in the Paris Brain Institute in France, and I've been mostly working on this topic about brain-heart interactions, and especially on clinical cases to understand what happened to consciousness and these brain-heart interactions when we have brain damage. So this whole line of research of brain-heart interactions comes from these ideas that have been pushed for a while that to try to understand consciousness and the mechanisms from a large scale perspective. This means, for instance, has been proposed for many years already, is like to revising some of the conceptions that traditional neuroscience has, like typically looking for brain activations for a specific context, but instead looking for what are the computations behind, what are the interactions, rather than specific region activations. And of course, when I said large scale, I'm talking about the whole system, the whole nervous system, or also considering the neurons that are outside the brain in other organs that have a lot to tell in many of these issues that we will discuss. As was anticipated already, why the heart and not other organ? Because why not, for instance, the spleen, the gut, or lungs? Why the heart? Well, the heart is a very special organ. So first of all, the heart is very easy to measure non-invasively. You just need two electrodes, two sensors. You place them non-invasively, and it's quite simple, and it's easy to know if you have a good quality signal or not. But more importantly, the heart is functionally connected to the sympathetic and parasympathetic nervous systems, and these both systems are directly or indirectly connected to all the body. So this is the heart is a good candidate as a proxy to what is happening in the rest of the body. And how does it work? The heart is it has this atrial node that contains these pacemaker cells that makes our heart beats, and the physiological mechanisms that is that we have sympathetic activations that cause the release of adrenaline that triggers the increase of these intracellular sodium levels that cause in practice increasing your heart rate, but also increasing the slow fluctuations of heart rate variability. So the changes in this and the heart beats around this baseline heart rate that is also increasing. And in the parasympathetic side, we have this release of acetylcholine that causes the inverse of the adrenaline. So it triggers this release of potassium in the extracellular space, and which causes decrease in heart rate and an increase in the fast fluctuations in heart rate variability. So looking at heart rate and the slow and fast fluctuations in heart rate variability is we can have a more or less an idea if there is a sympathetic or parasympathetic activation. So the brain and the heart communicate through different mechanisms that it's part of the family of what we call interoceptive mechanisms. And here I set between parentheses unconscious interoceptive mechanisms, because we mostly associate the word interoception to conscious interoception. So which is all this research that has been done in the field of psychology mainly, that is based on whether we can feel our heartbeats by just paying attention to our bodily sensations, whether we can feel our beating faster or stronger. But here we'll talk about unconscious mechanisms of what is happening in the physiological mechanisms behind, and that we are not aware of them. So we have so far molecular evidence, but also multisystemic. And here I have a nice figure that is an example for stroke. So stroke is where we have more evidence about how the brain and the heart are communicating. We have more evidence about the different mechanisms, as I said, from molecular, from multisystemic. For instance, well, in the most in the smaller scale, we have these microvesicles that are just these vesicles that go from to the extracellular space and travel through our system and reaches all the organs. It's known that stroke causes the release of these vesicles that contains some messages, for instance, in microRNAs that are basically just a mechanism that the cells have to regulate certain genetic expression. And this in part explain, for instance, some recent studies showing that genetic risk for cardiovascular diseases also happen to be related to a genetic risk for psychiatric disorders, and this has been somehow a hallmark in the last year on the field, where showing this suggesting this direct connection. But then we have other pathways like inflammation and all the neuroimmunological mechanisms that these two organs communicate. Then we have all the gut-related mechanisms that is in an indirect way, but it happens that the microbiota and it has also triggering some molecular mechanisms that can affect both the brain and the heart, and then we have all the neural mechanisms that we are mostly studying, as I said before, how the sympathetic and parasympathetic are serving as a pathway of communication between the brain and other organs. So if you pay attention to the figure, this figure has quite a few years. You see all the arrows going from the brain to the heart, with this conception that the brain is controlling everything. But we already have enough evidence to state that these pathways are bidirectional. So it's not the brain controlling everything, but also the heart is sending information and triggering some changes in the brain at all of these scales. So my proposal has been to, let's look at these large-scale physiological networks and with a specific protagonism to the heart, with the hypothesis, because it is still an hypothesis, that whatever happens to your body somehow will be reflected in your changes in the cardiac activity. And sooner or later this information will arrive to the brain, and it will react to these inputs. And I've been pushing this line of research to study brain-heart interactions and in different contexts. So in one hand, the topic of today, so it's basically critical care patients. So what happens to these kind of interactions in these different contexts, from brain death to brain damage that can suppress one specific function, such as the locked-in syndrome, that the person is fully aware but it cannot move because of these motor problems, but also in healthy participants to understand multisensory integration and sensory motor transformations. And brain-heart interactions have a lot to say about it. In the specific domain of critical care and disorders of consciousness, we have this typical classification with these two dimensions that to characterize consciousness in the in the x-axis a level of wakefulness, and the y-axis the level of awareness. So we see here that's obviously a healthy consciousness on wakefulness is on the top of everything, so it has higher of the two dimensions. But then when we go down, especially here for a minimally conscious and vegetative state, or on the unresponsive wakefulness syndrome, we have some overlap, because the distinction between these two is kind of challenging, especially because it's better explained here. So as I said, we have normal consciousness higher these two dimensions, but when we go to vegetative state, okay, we have this high level of arousal or wakefulness, but awareness goes very low and not necessarily zero. And then when we go to the minimally conscious states, this red line shows that, well, we have fluctuations in this level of awareness. So sometimes it might be easy to know the difference from a vegetative state, and sometimes not, and can lead to misdiagnosis, because there might be covert consciousness in the patient, and we cannot know from typical behavioral assessment, which is the standard approach to diagnose these patients in the clinical practice, just observing them through a method that is the coma recovery scale. And this is what is somehow motivating the exploration of the different brain imaging approaches and multimodal approaches to try to find this covert consciousness in these patients. So to exemplify how challenges is this distinction between the vegetative state and the minimally conscious state, I like to show this case, which is Vincent Lambert, that happened here in France. So he had an accident with a traumatic brain injury, and obviously was transferred to the ICU. This happened in 2008, and of course he never properly recovered after the coma, and was diagnosed in Belgium. He was transferred to Belgium for a better treatment and diagnosis, and he was diagnosed as minimally conscious state plus. So this means that Vincent was showing behavioral signs of consciousness that weren't just a reflex. There was a conscious contact in his reactions to different stimulations. But then he came back home in France, and across the years, the diagnosis was switching to likely unconscious, so vegetative state, and then since was then believed to be unconscious, some parts of the family started to request whether it's better for Vincent to receive this assisted death, which was finally permitted by 2019. So this shows how difficult is this case of consciousness disorders, because the medical teams have to make a decision on how to proceed with the patient, and whether the patient is conscious or not is very important for this decision. So in a study I will present you that was published in Journal of Neuroscience. So the question was whether looking at brain-heart interactions can serve as a marker to distinguish whether patients are conscious or unconscious after a coma stage caused by some brain damage. And this was a collaboration with the Coma Science Group in Liège. while I was working at the École Normale Supérieure in Paris. So we have a cohort of both minimally conscious and vegetative states or unresponsive patients. These patients, the analysis we did was mostly EEG, so bedside EEG without kind of manipulation or stimuli presented. And these patients were evaluated with two methods. So the standard behavioral assessment, which is the coma recovery scale, which is just an observation of the patient through a few weeks, and through a given score, the diagnosis is given either vegetative state or minimally conscious. And they also received this PET scan to see the preservation of the metabolic activity in the brain. And Liège has designed this approach to diagnose consciousness based on this method as well, only based on the metabolic activity of the brain. So given that we have a method that is based for diagnosis based only on behavior and another one only on brain imaging, you can imagine that there are mismatches. And the mismatch when a patient is in the behavioral assessment is considered vegetative states, but in the brain metabolism assessment is minimally conscious. These patients in between that are usually in the literature referred as minimally conscious state star, so MCS star, are also called non-behavioral minimally conscious. So given that we have these two kind of diagnosis of these patients, we could do some further analysis about this mismatch. So in the first part of the analysis, we focus only on the PET scan diagnosis. So we consider as a ground truth the brain imaging approach to classify these patients. And the main question was whether this method called heartbeat evoke response can detect residual consciousness. So the heartbeat evoke response is I will say, one of the most used methods to assess brain-heart interactions, and it's quite simple. It's just looking at what happens to the brain right after a heart beats. So it's the transient response of the brain to heartbeats, and these epochs of brain activity followed by each heartbeat are typically average, and is analyzed as an event-related potential. So this cohort has 68 patients, so both including both diagnoses, and we started with some machine learning. So we use an ensemble method of ensemble classifier, where the input to the classifier were the average of these responses to heartbeats and the variance of these responses. So the method here was what is called a random forest. Random forest might be already for the machine learning people an old-fashioned method as of today, but I still like it a lot because it's quite robust for this kind of data. So random forest is a bunch of decision trees, and in this case we use 1,000 decision trees, and each of these decision trees uses just a few features, just a few of them. And the method is maximized to make that each of the trees uses as different as possible features as inputs. And each of these trees will make independently by themselves a decision, in this case, whether it's minimally conscious of vegetative state, each patient. And the final decision is based on majority vote. So in this case, if we have 501 trees saying the patient is minimally conscious, that will be the diagnosis. And here we also, given that we know the answer of each of these trees, we computed what we call a consciousness score, which is just a proportion of trees that gave the majority vote. So this will be from 0 to 1. So in the case, for instance, that, for instance, a patient was diagnosed as minimally conscious with 501 trees supporting this, this will be a very, very low certainty score about the diagnosis. So we did a blind validation. So from the 68, half of them were we knew the diagnosis, we built the classifier, and the other half we in Paris, we didn't have idea of what was the diagnosis. So, and then we made our predictions, and this was sent to the team in Liège, and they gave us the accuracy, which was 87, which by the time was quite good. Was somehow similar to what is reached by using fMRI, for instance, with a huge difference that EEG is much cheaper than an fMRI. And we did several controls to know, to be sure that this was not a random finding, that this wasn't because matching parameters to make it work and everything. So one of the most robust controls we had was that locking these EEG epochs to the heartbeat. So looking like this time resolved analysis based on the cardiac cycle was always higher the accuracy when we were taking like surrogate heartbeats, random timings, as you see here in the figure, where we take epochs like not related to the heartbeat, and it was quite robust or method to always have a high accuracy, or most of the time have a high accuracy when was time resolved to the cardiac cycle. So this was really supporting the idea that looking at this time, these brain-heart interactions, we have this additional information. Because I have to say, when we were not locking to the heartbeat, these EEG epochs, we were still having a reasonable accuracy. It was just slightly higher when we were doing it based on the cardiac cycle. And then we explore which is the time window that is more informative with respect to the R peak of the cardiac cycle. So time 0 here, what you see in the x-axis is the latency with respect to the heartbeats, and then the y-axis is the relative accuracy. So we see that the highest accuracy is reached in the time window 200 to 400 milliseconds after the heartbeat, which shows which was somehow expected, that is somehow not exactly next to the heartbeat, but is in a later time window. And we also explore with the random forest the relevance of the different features that we were used for classifying, and we found that what was more informative was the variance. Instead of the average of these EEG epochs. So meaning that the variability of how the brain is responding to heartbeats is higher when the patient is conscious. And we had a slightly, I would say slightly, lateralization towards the right hemisphere, although the classifier is really using the whole scalp information. We did some tests by just using these few channels that you see more with higher relevance, and we didn't reach this 87 accuracy. So it was really most of the scalp was used to it. So, but with this slight right hemisphere trend. Well, as I said before, we had these MCS star patients that are diagnosed as vegetative state in the behavioral assessment, but as minimally conscious with the brain imaging, the PET analysis. And we had a super nice finding about this. So what you see here is the consciousness score that was given this random forest. So when the consciousness score was from 0.5, higher than 0.5 until 1, was diagnosed as by the classifier, but as minimally conscious. And we found that all these patients that are MCS star, well, not all of them, most of them that you see here as a star, had a lower consciousness score. So the classifier was saying these patients are minimally conscious, but I'm not so certain about it. So this was quite nice to find, because that this is suggesting that what the classifier is capturing in somehow it is what we call level of consciousness, but also detecting covert consciousness, which is really useful to diagnose, is potentially very useful to diagnose these non-behavioral minimally conscious patients. And we also, in parallel, we trained a similar classifier, but looking only at hard features, not hardware variability features, not putting any brain-related feature. And we also built this consciousness score that was from 0 to 1. And what you see here is the comparison of the consciousness score based on the heartbeat evoked potential, brain-heart interaction, and heart rate variability in the y-axis. And what is very interesting here is that this suggests that these two approaches are complementary, although heartbeat evoked response was always higher as well, that the classifier using heart rate variability. But what we see here that when either when the two of the scores in the two classifiers were very high, the classifiers combined somehow were making very good decisions to detect consciousness. And if you see all these blue dots are really like close to the threshold. So it's really suggesting, okay, what we are measuring with heartbeat evoked responses is not a cardiac only. So it's really an interaction, because it's not something that is not captured by heart rate variability. And another interesting thing we found, given that we have this metabolic brain imaging, we saw whether this consciousness score was related to metabolism in any part of the brain. And we have two small clusters that were well correlated with this consciousness score. So the first one in the ventral occipitotemporal region. So this is a region that is part of the ventral visual system, so face recognition, object recognition. And the second cluster was in the anterior superior temporal sulcus, which is is a small node of the full network, and is especially associated to social interaction and also self-related cognition as all is all the default network, but specifically this one on social interactions. A nice match that we have here is also right lateralized. So same as we found in the EEG analysis. So was a nice coincidence, I would say. So you might be wondering how this heartbeat evoked response looks. So in the vegetative state or unresponsive patient it looked mostly, not all the time I have to say, but mostly at the group level as just fluctuations around 0. While in the minimally conscious, we have some potentials that in this group, it was it looked negative, this potential that we see, But I have to say, not in all patients it looked like this, and some of them is actually, you see the same but positive. Uh, so the habit evoke response amplitude is not what is useful for the classifier. So it's just to be, if there is a presence of this response, that was most likely that what the classifier was trying detecting, and the variability of this response. So for interim conclusions, so first of all, habitable responses provide useful information to detect consciousness. And high variability is complementary to it. So how this happens, and why? So these are very broad questions, because first of all, it's machine learning. So even though with random forest allows to somehow decode what is happening, it is still a partial black box. So this motivated somehow this follow-up study, where we try to study in more detail what is happening in the habitable response in the different disorders of consciousness. Here we have a much smaller cohort, but with very clean data. So this allows us to do some better data mining in these patients. So we have 1 brain death, 6 coma, 8 vegetative states, 6 minimally conscious, 1 locked in. Locked in is totally conscious, and 6 controls. So this was resting states, e.g., like before, but in here we only have this standard behavioral assessment. We didn't have a PT diagnosis for this cohort. So first, what I want to show you, this is just a qualitative analysis, how one single subject of each of these categories looks like. So in blue, you see the habit evoke response. So the 0 is the heartbeat. And in the green is a surrogate. So random timing averaging many epochs of the EEG. So first, what you see in the conscious groups, somehow you have this positivity in the habit evoke response, both in the healthy and the locked-in syndrome. While looking random epochs, you just have some frequency activity that matches the average of the random epochs. So there is a clear distinction, somehow visually, at least, that is the information provided there by the habitable response. Then when we go to on the other extreme, the unconscious group, we have in the coma and the brain death something that looks rather flat. And what is important here, that if you look at the shaded areas of the plots, that is the variability of the group of all the epochs. In coma and brain death, there is no variability, so it's really disappeared, this shaded area. But then when we go to the problematic groups, that is the minimally conscious and the vegetative or unresponsive patients, is less straightforward, the difference, although there are differences. So the minimally conscious, first you see that there is something that we might think could be a habitable response. And then when we go to the vegetative states, is more unclear, although could be as well, there is a response there. But then when we go to the EEG non-locked to the heartbeat, so the green one, in the vegetative state looks like the same somehow, these two, although in the minimally conscious you could see, okay, could be that is slightly different. So, okay, this is just suggesting something, but of course, we cannot use this qualitative analysis to diagnose the patients. So that's why we went to quantitative analysis, motivated by these differences. So first here, I just want to show you that one of the challenges in all kind of event-related potential analysis in unresponsive patients is that the more severe is the patient, the more delta waves they have in the EEG. And delta waves is not it cannot be used as a marker of consciousness, because it's known already that it is possible to have consciousness in presence of high delta waves. So as you see here in the two power spectral densities, the unresponsive patient has much higher delta activity in the lower frequency range than minimally conscious. And this is just a result that when the brain is severely damaged, we have a natural slowing down of the brain activity, which results in a massive reduced activity in the higher frequency and an increase in the lower frequencies. And well, this is discussed in this paper, for instance, where they discuss about this paradox, like unconscious patients have high delta, but it's possible also to find consciousness with high delta. So it's a paradox. So this cannot be used solely as a hallmark to distinguish these patients. So we explore different features of the habitable response. So first, variance. As the first study I showed you, variability was informative. Here it is somehow as well. So for the test here, given that we had very small amount of patients, we pulled together brain death, coma, and vegetative states against minimally conscious, locked in, and healthy. So we have these two big groups as an initial analysis. So we had some differences in the variance here in this cohort as well, as you see here, and also in complexity. So this was L-seat complexity. Also, we found some differences between these two big groups, although when we look at differences in vegetative states and minimally conscious, it's not so evident, the difference. Then we explore segregation. And so segregation here, we use a network metric that is called local efficiency. Local efficiency is not a measure of segregation. It's, I would say, an indirect way to measure segregation, but in this case, it is segregation, because we found that there was a high local efficiency in midline frontal channel as compared to all the rest of the scalp. So that means that in frontal electrodes, there is a high correlation in all these neighboring channels on the habitable response as compared to the other channels. So there is a focused response in the frontal electrodes in this cohort at least. And this was also somehow separating the big unconscious from the big conscious group. And then we look at the specificity, so that that's a measure that is a measure based on wavelength entropies, that is just a comparison how this habitable, the amount of information, I would say, of the habitable response as compared to the surrogates. So we compute several surrogates, like random timings of the heartbeats, and we see how much information changes we take a measure of distance, which is this specificity towards the surrogates. And it's also somehow separating these two big groups that we defined. So the question is there redundancy about these four features we found? So we just did a simple correlation analysis to see whether if these measures are complementary or not. And we found that the ones that are the least correlated are this measure of this frontal segregation and the variance. So this was the not correlated at all. So this is how these two measures look as plotted in a two-dimensional space. So what we see is the more conscious is the patient, the farther is from the origin in this plot, accounting from habitable response variance and segregation. And if we just draw a separation between these two, we see that most of these two big groups can be separated, although we have three patients misclassified, if we can call it like this, by just looking at these two features. So then this would motivate us to look, okay, let's see if heart rate variability and habit evoked response remain complementary like it happened in the last study. And so we combined the habit evoked response variance and segregation in a single measure, and we plotted against heart rate variability high frequency. And here in this plot, we totally separate the vegetatives from the minimally conscious. So a nice result confirming what we found in the previous study, this complementarity between these two kind of approaches. Although it's important to say that heart rate variability is not yet a hallmark marker to detect consciousness, if you do a quick search, heart rate variability source of consciousness, you will find many papers. I named some of them here, which is included in my study, the first one I present you, because we also made a classifier based on heart rate variability. So most of them suggest, okay, heart rate variability is somehow useful, but it's not yet completely separating the patient groups. The results we see is only trends, the trends that certain aspects of heart rate variability in certain contexts could provide information. Which is fine. So, which is also good, nice that we see that it's complementary to the habit evoked response. So, entering conclusions here, we have, well, we validated somehow the previous conclusions of the previous study that the habit evoked response can provide information about consciousness, and that heart rate variability can serve as a complementary information. So what about presenting stimuli to the patients? Because all these studies were in resting state. Well, resting state, we can call it like this, is bedside EEG. So in a follow-up study, we studied the patients, a different cohort that was from Paris, 40 vegetative states and 46 minimally conscious states, and in addition to 11 controls. These patients had this EEG under auditory stimuli and were assessed by the standard behavioral assessment. So here we wanted to see whether this auditory information and perception is somehow differentiated in these brain-heart interactions in these patients. And this was motivated because we have already a lot of evidence in healthy participants that the different approaches of analyzing perception, detection of stimuli, and is somehow related to the cardiac cycle. And this has been shown already in auditory, tactile, and visual, but I'm just naming a few studies, but there are more. And these studies follow either two different approaches, whether to check if the stimuli fall in during the systole or diastole of the cardiac cycle. Systole is the time window that is around the heartbeat, because it's related to the contraction, and diastole is related to the relaxation of the heartbeat. So before the heart will beat again. And all the other approaches, as I told you before, this habit evoked response, so looking at the heart brain activity locked to the heartbeat or the cardiac cycle. So here, in particular, the kind of auditory stimuli we had was the local global paradigm, which is a sequence of 5 sounds that where the fifth is either the same as before or different. So you have a beep, 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 beep. And this deviation can happen at a local short-term level or global longer-term. And we analyze neural activity based on this to see the response to this fifth sound. So first, we did this typical event-related potential analysis to look to EEG activity locked to the onset of the fifth sound. And the other approach we used was to look at the heartbeat right after the sound, the fifth sound. What was the heartbeat evoke response associated? So we have these two type of markers at both levels: local auditory irregularities or global. So the main result we found first was that EEG locked to sounds produced this frontal event-related potential that was higher in healthy control and slightly higher in minimally conscious as compared to the unresponsive patients, which was as expected. This has been shown already in the past. And then when we go to the EEG locked to the heartbeats after the fifth sound, we found effects at both global and local levels. So in the global, we found around the same time window, that's the local effects. We found a higher response in controls and minimally conscious. And then for the local level, we found a more dynamic response that originates in central right areas and then somehow travels to the frontal towards the left hemisphere, which was also higher in controls and minimally conscious and slightly lower in unresponsive. We did several controls here as well to see whether is really needed the heartbeat for this, and we confirm it. We also confirmed that these three effects were uncorrelated, so also providing between them complementary information, which was a nice result that lead us to the conclusion that all other people have reached the same conclusion as well, like in different paradigms, but interoceptive, exteroceptive interactions. So what happens inside our bodies while we are processing the external world somehow is relevant for perceiving the world, and here for detecting presence of consciousness. As a next study, I have here a case report where we also wanted to study the heartbeat evoke response. This case report was a woman that was suffering ventricular tachycardia. We have the recording up for 20 hours before she passed. So that's why we call it a near death case report, because we could capture the hours before she passed away, unfortunately. And so I will briefly explain you that, well, what happened here. So the woman developed ventricular tachycardia that ended up being ventricular fibrillation. So in simple words, what is happening to the heart here is that is beating out of control, so beating very fast, and which doesn't give enough time to refill with blood. So the heart keeps beating without having enough blood to pump towards the rest of the body, specifically the brain, and this leads to severe brain damage. And the ventricular fibrillation evolution is characterized by these changes in the patterns of the electrocardiogram. So what you see first is something that looks more or less what would be a normal sinus, but then you see this kind of patterns, like during the tachycardia, and the final fibrillation, where just the heart is oscillating, not even beating. So what we explored first was, how does it look, this heartbeat evoke response during the normal sinus, or more or less normal sinus. And we see this, here you see how it looks on time in different EEG channels. But here you see as well how it looks at scalp level at different time windows with respect to the heartbeat. So what we repeatedly found in different time windows, here I'm just showing you two different time windows, is this somehow midline positivity of the habitable response between 0.1 and 0.2 seconds from respect to heartbeat, which is we cannot say that is good or bad, but it's there. But when the tachycardia evolved, even at the very early stages, this response disappears, so it goes towards zero. So here we can say at least measuring the brain interaction through heartbeatable responses, we can say, okay, the brain is not responding to individual heartbeats, which was expected. But then when we look to heart rate variability, so how the heart rate oscillates on time. So you see here two different time windows. You see on top the pattern of the electrocardiogram. You see down here a 1 minute time window, how it changes of time, the different rhythms of the heart rate. So in black is the interbeat interval, so it's the inverse of the heart rate. So you can see it as a heart rate measure. And then low frequency, high frequency, and very high frequency. So slow to very fast oscillations of heart rate. And we see that we have an increase in high and very high oscillations around 10 seconds, which is followed by an increase in the EEG side about the whole spectrum. There is a increase in EEG activity, and then it's followed by an increase in these rhythms in the heart, and which is somehow then in the second example is somehow inverse. We see this increase in cardiac activity, and then it's followed by an increase in the EEG activity in a broader spectrum from delta to the gamma bands. So this we could call it somehow aberrant coupling, and it's happening by time to time it was happening by time to time in this patient. So we could say that sometimes cardiac oscillation and brain oscillations are very coupled. So as an interim conclusion, so first this near-death case report did not have responses to individual heartbeats, but there was a suggestion of aberrant coupling in certain time windows between EEG oscillations and heart rate variability. Of course, for having a stronger conclusion, we need a larger cohort. So this motivated the next study, where we have many patients. So it was 181 comatose post-cardiac arrest patients that developed encephalopathy and were in a state of coma. From this cohort, we have some patients with a good outcome. So that means that survive. And then, but most of them, unfortunately, did not. So that we label as poor outcome. This was bedside EEG. At the start of the coma stage, we had EEG and ECG, and they were evaluated in the ICU with typical traditional prognostication markers that I will explain you later on. So here what we did was a little bit more complicated. We did what I call some reverse engineering of brain and heart oscillations. I will go explain you briefly by the different blocks. So here we have what we can measure non-invasively, the electrocardiogram and the EEG signals. So we know from physiological modeling literature that we can model how the heartbeats are generated. So it's this red block, which is basically the stimulation of the sinoatrial node that has these pacemaker cells that I explained you at the very beginning of the talk. And these are stimulated by this autonomic information inputs. And this autonomic information that is stimulating this sinoatrial node are composed by, here in the yellow block, by a sympathetic and parasympathetic activity that are combined. And this sympathetic and parasympathetic activity here we assume are somehow modulated by the brain. So this is the blue arrow that we see here. And on the brain side, we know that the EEG is composed as different frequencies. Here we have the strong assumption that we can model separately each of these frequency bands. I know that there is evidence that these bands interact between each other, because for instance, we can have a neural process that propagates first a certain frequency and then changes. But here we model it for sake of simplicity as separate mechanisms, delta, theta, alpha, beta, and gamma. And we model independently how both sympathetic and parasympathetic activity is modulating the power of each of these frequency bands. So we have all this model couple, which results in summary time-varying markers. So we obtain a time-varying markers from brain to sympathetic, brain to parasympathetic, sympathetic to brain, parasympathetic to brain. We get these 4 time courses, as you see here in the figure, for each EEG channel and each EEG band. So a bunch of markers. So in this cohort, given that in ICU patients, very high frequency EEG disappears or almost disappears, we focus on only low frequencies, so between the delta and the alpha range, so up to 12 hertz. And the first finding, we found that some measures of brain heart coupling were linked to the qualitative EEG analysis. So in the ICU, qualitative EEG analysis is traditional practice, where the specialists just look how the EEG looks, and then it's classified as highly malignant, and benign. And we found that this subjective classification given by the specialist was correlated with or somehow related to our brain heart coupling. So the trend for alpha, theta, and delta were very similar. Here I'm just showing you one example. Here was the alpha to low frequency and alpha to high frequency heart rate variability. So here we found that the highly malignant patients had higher alpha to high frequency compared to the benign that was slightly lower. And we found somehow the inverse when we looked to alpha to low frequency, but the effect was much stronger in the alpha to high frequency in this case. So first result, we somehow capture these subjective reports or part of it. Another result that we found that brain heart coupling was linked to the presence of somatory evoked potentials. So somatosensory evoked potentials are used in the clinical practice as a binary marker of prognosis. So whether they do the somatosensory stimulation and they see in the EEG the potential, the response to it is likely to survive, and if not, is unlikely to survive. And we also saw that whether the presence of this marker was also related to the different levels of brain heart coupling. And if you see here at the bottom, in this case, for instance, delta to high frequency, so brain to high frequency heart rate variability was higher in the patients that did not show the presence of this somatosensory evoked potential, so correlating with another marker of prognosis. And another result that we found, that another marker used in the clinical practice for prognosis is the neuron specific enolase. Enolase is an enzyme. It's neuron-specific, but it's an enzyme. Like all enzymes they have a role in metabolism, and it happens when we have brain damage. The levels of this enzyme increase. So that's why it's used as a marker of how severe and is the patient. And we found a nice correlation with this concentration of this marker at the third day. And we see here again, well, in this case is theta, the example, towards the high frequency heart variability, and we show the higher concentration of the enzyme, which is indicated of that the patient is really bad, also related to very high coupling in these two components. So here I just showing you an example how a good outcome patient looks. So on top we have the time course of the EEG power in the alpha band. We cannot say much about it. There is activity. Then we see in this example is low frequency heart rate variability. Same, there is variability. And in the green one we see the time varying, the time resolved coupling estimation, which is, well, we don't know with a simple view what is happening here. Could be coupling, could be not. Most probably yes. But when we see a poor outcome, this is how it looks. We see suddenly a huge peak in this case of alpha activity, and then this happened somehow in parallel with this peak in heart rate variability. So this is really interesting because the in the critical care patients overall, the more severe is the patient, the lower variability you find. This is a trend, obviously. It's not always like this, but it's typical finding that there is no variability, and it makes sense. Like someone having a constant heart rate is clearly a sign that something is not very well. We need changes in the heart rate, and we need to have some heart rate variability because it's part of regulation processes of our body is doing on real time. And here we have this massive coupling, and this is captured as the model as super couple, aberrant coupling. And so here we can say, so poor outcome was associated with highly coupled, but we also see, given that it's just a single peak, highly coupled is low complexity, and we test it, and we show that there is a trend that highly coupled and low complexity are both associated to poor outcome in the brain-heart interactions. So interim conclusion, aberrant coupling between EEG and heart rate variability is linked for both severity and prognosis in post-cardiac arrest patients who suffered encephalopathy. And this is my last study, is not published yet, will be preprint soon, I hope. So we did a similar analysis that the previous study, so modeling these brain-heart interactions, but in this case was COVID patients that were in the ICU. So we have 52 patients, all of them from 2020. They were under deep sedation, and well, COVID, of course, was confirmed with a PCR, and we also had EEG and ECG in all these two patients. And we had the data available for 2 time points of the patients. So T1 associated to deep sedation, and T2 shortly after no sedation. So we have 2 different conditions to compare. So the first results, very, very robust, was the changes in the sedation. So with and without sedation changes the brain-heart coupling, which is also, I mean, why not? It's not unexpected. So here you see brain to heart. So CPI is cardiac parasympathetic index in this study. We see an increase in the brain to heart after sedation weaning, so without sedation. And then it's an inverse effect. So heart to brain when without sedation is reduced. And but if you look in detail these two figures, there are some patients that do not follow this trend. Some patients, yes, but some not. So this motivates us to look further what is going on behind. So we did a median split of the coma duration. So we have patients with short coma duration and long coma duration. So the coma was from 0 to, in this cohort, from almost 2 months. And we look at the same markers of these figures. So how the distribution looks. So on top in the two, you see the patients that were really severe, so with a longer coma. And at the bottom, you see the patients with a shorter coma that were better. And what you see, the difference in the when you stop the sedation, is what you see on top is that the very severe patients do not follow a strong change in the brain-heart interactions in both directions, especially in the heart to brain. The median is almost the same. But then when you see the patients that are not so severe, you have a stronger modulation of the brain-heart interactions. So that means that suggests that the amplitude of this brain-heart coupling is not the most important, but how it changes, how these brain-heart interactions dynamically change based on the conditions that you present. And the better the patient is, the more dynamic is this change in the brain-heart interaction. Another nice finding we find in this cohort is that we found that this brain-heart coupling was related to the severity measured as the coma duration. So we found a nice correlation here with the coma duration in days, but also in the coma-free duration. So it's the second column in a 1-month period. So again, stronger brain to heart coupling, the more severe the patient is. And more interesting for me, at least, is because it's directly related to consciousness, is we also have the information about delirium days. So delirium is this state of the patient that is characterized by disorientation, confusion, but especially lack of awareness of what is going on. So obviously this is evaluated like subjectively by the specialist, but we have a marker of whether the number of days the patient during the hospitalization had these stages, and so the higher the number of delirium days, the more severe. So also made sense. And then here I just have just to show you how it looks at a patient who spent many days in coma and a patient who spent just a few days. So in the patient who spend many days in the coma, we always find higher coupling, and it's partially explained because there is a slowing down of the brain activity, as you see here in this EEG power from delta to alpha band. You see this is slowing down, so the changes in the brain activity is very slow and somehow in synchrony with the heart activity. And then we see that on the right, a patient that is not so severe, and you see like faster changes in the EEG power that are less likely to be coupled with the heart. So this is more or less what is happening here. Still, these are preliminary results. We are still controlling the potential presence of confounds that could be. In regarding this relationship of brain-heart interactions with delirium opens a whole new window of possibilities of studying altered states of consciousness in this with these measures of brain-heart interactions, because delirium in catatonia have closely related, although catatonia is not found in the ICU, not necessarily. It's more like psychiatric associated to psychiatric conditions, and it's more related to behavior. rather than awareness, but still it could be potentially considered as an altered state of consciousness, which will be super interesting to continue delving into it. So as an interim conclusion about the COVID cohort, so again, brain heart coupling is linked to the severity and also this delirium. We did not find relationships on prognosis like the previous cohorts. In the previous cohort, we somehow could predict whether the patient will survive. Here was not the case, also because we had a much smaller cohort. So final conclusions: brain-heart interactions are potential biomarkers of presence of consciousness, covert consciousness, as we saw in the non-behavioral minimally conscious states, level of consciousness, severity after brain damage, prognosis after brain damage, and potentially for altered state of consciousness, which was the case of delirium. And here just a closing open question. So why this brain-heart interactions agenda, I believe, is quite important, is to answer these kind of questions, whether if we can keep our brain alive outside the rest of the body, whether we be conscious, or whether can we make conscious these lab-grown brains, organoids, without other organs, just by supplying it the minimum nutritional requirements and atmospheric conditions. So here, acknowledgement for all the people and institutions that have supported all the papers I presented you. There are many. So thanks to all the institutions and the funders, and of course the amazing co-authors that are all listed them. And last but not least, thanks to the patients and the proxies to giving permission for following all these super interesting and necessary research. And that was my last slide.